Hello and welcome to section 5 of your video lecture on the digestive system. In this video, we are going to discuss the small and large intestines. So we'll start off with the small intestines. This is where the greatest amount of digestion and absorption of nutrients and water will take place. Altogether, it is approximately 3 meters long or 10 feet long, which is twice as long in a cadaver. And our surface area is going to be 100 times the surface area of our skin. And that's going to be due to a couple folds that we'll look at when we see the histology of the small intestines. We divide it into 3 different parts. We've got the duodenum, which is the shortest, the jejunum, and the ileum. Now, our duodenum is going to be the first 10 inches beyond the pyloric sphincter and is a retroperitoneal structure. You can see that it has this C shape to it and it cradles the head of the pancreas here. Now, the reason it does that is because we are going to have some structures entering into the second part or the descending part of the duodenum. We have this hepatopancreatic ampulla, also known as the ampulla of Varder. And that word ampulla just means that we have a widening of a tube here that is going to allow contents to move in from the liver and gallbladder, which is bile, to move into the duodenum, as well as a duct from the pancreas that allows pancreatic juices to empty into the duodenum. Now, what helps control the secretion of these fluids is going to be this muscle in here known as the hepatopancreatic sphincter or sphincter of Oddi. When it contracts, it closes up the lumen and won't allow bile or pancreatic juices to move in, but when it relaxes, it will open up the lumens and allow the bile and pancreatic juices to move in. The opening where these juices flow out of is called the major duodenal papilla. So this is a really great view in which we see the widening called the ampulla, the muscle, which is our hepatopancreatic sphincter, and then the opening, which is our major duodenal papilla. So next we'll move into the jejunum, which is about 3 feet long, and the word jejunum means empty, and the ileum, which is the longest part of the small intestines, which is approximately 6 feet long. We could see that the ileum is going to be thicker and more vascular with its blood supply here, and will join the cecum, being the first part of that colon, the large intestines, an area known as the ileocecal junction. And that is where we will also have the ileocecal sphincter or valve that will ensure that the contents of the ileum remain flowing in one direction into the cecum. Remember that the ileum here is also going to contain a special feature known as Peyer's patches, which are lymphatic nodules surveillancing for pathogens. So I said we were going to talk about why the small intestines has such a great surface area. It's going to have a 600-fold increase in its surface area, and that is going to be due to something called plicae circularis, also known as circular folds. So if you look inside the intestines here, we could see that the mucosa is folded in these circular folds. And then if we look closer, we have those finger-like projections known as villi. And here we will have the blood capillaries along with the lacteal, which is a lymphatic capillary. So this is further folding the mucosa. And then sitting on these columnar cells here, we are going to close up at one of those cells, and at the very top we have microvilli, which are also known as a brush border, and the microvilli help to increase surface area and serve as absorptive features of the cell. So more on the histology here, we do have intestinal glands, and these are going to be our tubular glands of the mucosa at the basis of the villi, and they help to produce intestinal juice that is going to be slightly alkaline, especially because we want to neutralize some of the acid, although I should say some of the acid that is coming from the stomach, that acidic chyme, although the pancreas does a lot of that for us in secreting an alkaline fluid that we'll get into later on. So our duodenal glands that we could see here are called our burner glands, and these are tubular mucus glands of the submucosa that open up into the intestinal glands. Here they're going to produce a bicarbonate-rich alkaline mucus that helps to buffer that acidic chyme as well. So let's take a look at some of those cells and glands within the mucosa. This is kind of similar to what we saw within the stomach, except instead of having the rugae and gastric pits, we now have villi. And along here, we are going to have our absorptive cells that you see in the light cream color. These are also known as enterocytes that will have the microvilli sitting on top of them, and they will produce digestive enzymes and absorb the digested food. Sitting in between here too, we also have some goblet cells, which I don't think are pictured. Oh yes, they are. They're represented by the dark brown cells here, and they are in charge of producing mucus to line the mucosa. And then deeper, we have our endocrine cells that are going to produce regulatory hormones. And lastly, our Paneth cells, which are the deepest. And these are also known as granular cells that help protect us from bacterial infections in the intestines. So let's go through, similar to the stomach, how we went through mechanical digestion and chemical digestion. So within the small intestines, the mechanical digestion that takes place is segmentation, in which we are moving that food back and forth in order to combine the chyme with the with the digestive juices. And then we are pushing the food against the mucosa so that we can absorb the nutrients within the food. And this takes about 90 to 120 minutes to reach the end of the ileum, which we see over here. Then that motility will increase in the ileum so that we can move through the ileocecal valve and move that food into the large intestines, specifically at the cecum. As far as chemical digestion, we'll have our proteins and carbs completely digested here through the intestinal and pancreatic juices. We focus on lipid digestion because within that villi, we do have that lacteal. First, in the duodenum, the bile is going to emulsify the fat, and then from the pancreatic juices, we release a pancreatic lipase, which will then further digest the fats. So eventually we will end up with monoglycerides and fatty acids. We also will absorb water here in the small intestines through osmosis. And optimal digestion is when that chyme is delivered slowly and in small amounts so we can ensure to uptake the majority of the nutrients within the food. Now let's switch gears and talk about the large intestines. The primary function here is to absorb nutrients and water, although most of that takes place in the small intestines. But we also want to synthesize certain vitamins, form the feces, and then eliminate that feces from the body. This large intestines will start from the cecum, which has the vermiform appendix on it, and will end at the anus. So notice how the large intestines is creating a frame around the small intestines on three sides, and then we'll move posteriorly to the anus. So let's talk about the different parts. First, we have that cecum, which is a blind sac that is suspended inferiorly from that ileocecal valve. And here we're receiving the contents from the ileum. Hanging off of that cecum is the vermiform appendix. That is a winding tube that attaches to the cecum and contains the lymphoid tissue, again serving as more surveillance for pathogens. And from there, we are going to move superiorly or up. There we are going to call this the ascending colon, and this is on the right side of the abdomen. So you can kind of follow along on your own abdomen, moving up the right side of your abdomen, and this is a retroperitoneal structure. then we are going to have a turn, and this turn takes place over by the liver. So in the upper right quadrant, we have our liver here, and that's why we call this the hepatic flexure, hepato meaning liver, or the right colic flexure, meaning on the right side of the colon, we are flexing, we are folding and moving in the opposite direction. So we will move from the right side of the abdomen now to the left side through what we call the transverse colon, and there we have another bend, and this time it is in the upper left quadrant where the spleen is, so we call this the splenic flexure or the left colic flexure. From there, we're going to move down the left side of the abdomen, and this is our descending colon. And once we hit a bend here, this loop of colon is called our sigmoid colon. I forgot to note that the descending colon is also retroperitoneal, just like the ascending colon. So the sigmoid colon is S-shaped, and that's why we say it is a loop, and it's entering the pelvis inferiorly. From there, of course, we then would have the colon move the contents into the rectum and then out the anus. Next, we have our little hemostatic imbalance known as colorectal cancer. This is one of the most deadly malignancies, and you are at risk for this type of cancer if you have a family history of it, are a smoker, have excessive alcohol consumption, and a diet that is high in animal fat and protein. Symptoms would include constipation and diarrhea, cramping, abdominal pain, and rectal bleeding, and it can arise from a benign mucosal growth called a polyp, and this is why as we get older we need to have colonoscopies so that we can identify any precancerous polyps and remove the polyp before it becomes malignant, and those are recommended for people who are 50 years old or older. So we ended up at the rectum when we last talked about the structure here. This too is a retroperitoneal structure that is going to course anterior to the sacrum and the coccyx and leads to the anal canal, which is located within the perineum. There's two sphincters here. We have a internal sphincter, which you see pictured in the blue here, and is made up of smooth muscle and is under involuntary control. And in the green here, external to the internal anal sphincter, is of course our external anal sphincter. And this one is made out of skeletal muscle and is under voluntary control. So typically this remains closed unless you voluntarily relax these muscles, specifically the external anal sphincter, and allow defecation to take place. Some other special features of the large intestines is that we have the circular layer of the musculus externa complete, but the longitudinal layer is incomplete. And so it creates these bands known as tennae coli. And by contracting the tennae coli, it forms these pouches on the large intestines known as haustra. Hanging off of the large intestines, we have these fat-filled pouches, which are called epiploic or omental appendages. The function of these are currently unknown. And if we look at the mucosa of the large intestines, we know that we will have numerous straight tubular glands called crypts, and we'll see a lot of goblet cells because we need to secrete the mucus in order to kind of compact and create the feces so it can smoothly move through the colon in order for it to exit the body. There are also going to be absorptive and granular cells, as we saw within the small intestines, here in the large intestines as well. So similar to what we did within the stomach and small intestines, let's talk about some of the digestive functions here. We do go through mechanical digestion using three movements. We have our haustral contraction, in which we get more of a sluggish type of segmentation when we have food in the colon. So it'll kind of contract just these little pouches here. And then we also get peristalsis, which is going to be slower, but we eventually are moving its way toward the rectum. And lastly, we have mass movements. Now, these are going to be strong waves that help force the contents toward the rectum. And we also have something called a gastrocolic reflex. This means when we have food within the stomach, this causes distension, pushing on those stretch receptors, and will cause motility within the colon. So I like this little cartoon here. Now for chemical digestion, we just have that mucus being secreted, but we don't have any enzymes to further break down the food. Only bacteria here in the large intestines will help with chemical digestion. So our feces here will be made up of food residues that are undigested. We didn't absorb these substances, and we also have bacteria or old epithelial cells that have sloughed off. We also have inorganic salts and water. Now, in order to go through the process of defecation, we can use a Valsalva's maneuver. This is a voluntary procedure in which we create intra-abdominal pressure by contracting our diaphragm and our abdominal muscles, and also a closing of your glottis. So we are going to use that false vocal fold that we talked about in the respiratory system to create the intra-abdominal pressure. And then we can have our defecation reflex take place. This means we have feces within the rectum that is stretching the wall, causing elimination of the feces. It moves into the anal canal and sends a signal to our brain, giving us the choice to use our voluntarily triggered external anal sphincter to relax in order to open up and allow the feces to move through.